So now we come to the last part of our series of biochemical characterization techniques, and we're going to talk about things which are related to both biochemistry and also databases and computational techniques and so on and so forth. So in this era, we are now in a position where we have so much data and so much ability to store the data that we can easily combine computational techniques and biochemistry. So things like the terms like omics, genomics, proteomics, these are very common terms now. We're going to look at what they mean. The rest of these terms, bioinformatics, computational biology, systems biology, synthetic biology, they are all beyond the scope of our syllabus. But as you can see, the omics and the bioinformatics, these are a lot of data which we can use, and they're basically huge databases which we put into our system. Say you use some sort of a program or some sort of a data computing software, and then you use it to figure out certain metabolic processes or lots of other things, which gives rise to the fields of systems biology and synthetic biology. Let's focus on the omics for our part of the syllabus. So you've all heard of the human genome process, the human genome project. That basically mapped every possible gene in the human being. And there are a lot of other genome projects going on. You have your RISE genomic project, JUICE genome project, so on and so forth. So what is genomics? Genomics is the compilation of all the data from all the genes of an organism or of all any organisms. And then you have proteomics and transcriptomics, which are the databases of RNA and proteins, and then others like lipomics, metabolomics. There are many omics, and basically these are big databases of all the biological organelles and biochemicals that we have seen, and those, as we've seen, give rise to they contribute to systems biology. A little more in detail. So we know what is a genome. A genome is the organism's complete set of DNA. So all of its genes, when you can compile it and put it in a database, that's the genome. Similarly, the proteome is every single protein that a organism possesses. So proteomics and genomics are then the whole methodology used to catalog the identity and function of these proteins and genes. Now coming to some examples of various databases. So you can have DNA databases, RNA databases, and genome databases, and on one hand, and you can have protein databases. With protein, you can have say databases for just the sequence. You can have databases for the structure. So you can basically primary structure, you can have separate databases, and structure based databases can be separate. We'll look at some examples of that. You can even have some databases which catalog protein interactions, and that is a little bit beyond our scope, and also these we won't be looking at, but it's nice to know there are other databases. For example, carbohydrate structure, PCR primer database, metabolic pathway database, and so on and so forth. So everything that now we journal, or once we figure out in the lab and then put it in a publication, you have another place to put that. You have these databases where you can put all that information, all that biological information that you have figured out in the lab. Okay, so just a little bit more. The DNA databases are these are the common databases. We'll look at protein databases as well. For example, GENBANK, ENA, DDBJ. Okay, and we'll look at what this REFSEQ means in a bit. So here are some the the names that I just said. Okay, for protein and we just looked at the genetic databases. Here are some protein databases like NCBI, EMBL, XPC, PDB, etc. And throughout this lecture, we'll also be looking at some tools to handle the information that you get from these. Okay, so there's a BLAST, FASTA, and other certain tools. Before going into the detailed examples, we are going to look at what type of databases you can have. There are two types basically, primary databases and derivative databases. Primary databases are basically original submissions. So anybody can actually make a primary data. You can just type in my grandma's genomic and then you can just put it in. Okay, so a good example is GenBank, and we'll be looking at it in detail. Some other databases are there that they take all these very primary data and then they curate it, or they correct it, or they compile it, and then they say it's a derivative database, right? So it's not the original, but it's more organized, and a lot of erroneous database can be discarded, a lot of repetitive data can be discarded. An example of that is REFSEQ. Both of them are NCBI owned databases. We look at that. Okay. So NCBI is basically, let's go to this NCBI site, ncbi.org. Right. I'm sorry. So this is basically an American-based, National Institute of Health-based database. It's called National Center for Biotechnology Information. So already you can see it's about bioinformatics, and you can have these databases. You can have these are all like different things, but what we are more interested in is gene, genome, nucleotide, protein clusters. These things we are going to look at for just with structure. Okay, so these over the years they've become very rich. Again, here you can also go for DNA, RNA, genomes, and proteins, sequence analysis. So this is basically a very good example of a very rich database. You can submit, you can download, and you can analyze, and lots of things. Let's see how we do a typical search. I'm going to just skip this, and I'm going to ask you to read up the slides that I have just to give you a comparison of why GenBank and REFSEQ are related but different. We are just going to go ahead and do a GenBank search. Okay, so for example, if I take this and paste it here, so this is my search parameter. I can just go ahead and just write myoglobin, and it will give me this. If I go for all databases, it's going to be very clunky. I'm just going to go and write nucleotides. So as you can see, it's given you a huge number of myoglobin related proteins. You have GenBank. Most of them is GenBank, right? So let's just pick one. Okay, and here the first one is the first two lines. It has a lot of data. It's since I looked at nucleotide, it's giving me the nucleotide information. It's linear, it's not circular. 723 base pairs. It's a DNA, it's not an RNA, and it's called melosome leukotype occipitalis voucher myoglobin, and this is an important number. This is the reference number for this particular protein. Okay, and whenever you want to access this, you can just put that in, and here is your organism. The organism is a little bit detailed over here, eukaryote and chordata, so it's basically a vertebrate myoglobin. There are other things over here. What I'm more interested is here. It's giving you the base sequence. So it's, for some reason it doesn't start with the ATG. I don't know why that is. Let's take another example. I'm just going to paste it in the search bar. Search, yeah, okay. So look at this. This is basically I think it's a warm myoglobin, and it's the same things. It'll give you the definition, the accession or the reference number, and the organism. And here, okay, this is important. Look here, this is saying interpro. This is an interesting piece of information because NCBI is not the only protein database in the world. That's American based, and there are these European database places, UniProt KB or SwissProt. Okay, so if I just write uniprot.com, no, uniprot.org, is it? And I take the number over here, P12844, P12844, and I give it to search. It's going to give me the same protein. Okay, and it's going to give me similar information. If I go into this, let me see if I can find it. Expression and all that thing. There you go. So this, let me see if I can zoom out. Okay, so MSGNPDAFEN, and we were doing the same thing, MSGS. So basically what I'm trying to say is that there are a lot of protein databases out there or nucleotide databases out of there. Obviously they are from different sources, but when you talk about the same protein, they should give you the same information. And not only that, it's nicely cross-referenced. For example, NCBI will give you a reference number for a different database, that is UniProt or SwissProt and stuff like that. So if you're looking for a particular protein and you want to say, okay, this is a very common protein, I want to figure out the primary sequence. Here you go. Nice enough of them to also put in the base pair sequence or the nucleotide sequence. Okay, so you have that both. Okay, and there are tools where you can just copy the base sequence, and it will give you the corresponding amino acid sequence. So that's about it. I'm going to just let's see if I missed out all the one. Okay, so I'm going to stop right here. That protein sequence we just looked at once, like REFSEQ or GENBANK. I should write GENBANK here. GENBANK or REFSEQ, and correspondingly you have SwissProt or UniProt, and there are lots of others. Okay, so go ahead and experiment with those, and we will look at some of the others in the next video.